And we don't have like this full hour of things to talk about because we want you guys to have plenty of time to ask questions. So feel free at any point, raise your hand so we can address your questions at the right time. You don't have to save your questions till the end, okay? So what we're going to do is give you a little background about horseshoe crabs in general. We're going to tell you what we think are some of the coolest things about horseshoe crabs. And of course that's going to touch a lot on our own work with horseshoe crabs. And then we're going to talk to you about what we know about the horseshoe crabs that live in Great Bay and their behavior in Great Bay. And we're going to ease into this spawning phenomenon that we're getting ready to study in May and June. We're also going to talk to you about why it's important that we do these surveys, because currently there's some concern about horseshoe crab populations all up and down the eastern seaboard, because for a couple of reasons their numbers have gone down. And so we're going to spend some time on that as well. Okay? So that's kind of the big picture. Okay, so let's go way back. Let's go back 450 million years. Based on the fossil data, they've been here for a long, long time. They clearly are very well adapted to living on this planet. And they're also very adaptable, flexible, because they live everywhere from Mexico up to Maine, right, in estuaries and in the ocean. So they're very hardy animals. And they can also live with very little oxygen. They'll bury in the mud and they can survive under those conditions as well. So they're very tough animals and they've been here for a long, long time. There's several different species. You guys are familiar with the ones that live on the east coast of the USA, but there are also some that are over in Asia as well. And that population is also in big trouble. So, as many of you know, from just walking around the beaches in May and June around here, all of a sudden, sometimes you'll just see like, 100 horseshoe crabs crawling up from the beach. You go down to Chesapeake Bay and stuff, it's crazy out of control. There's so many of these guys. And so, for years, they've been used for things as crazy as fertilizer, right? And who cares? There's a bazillion of them, and they're readily available and they're free, and let's just use them for that kind of stuff, right? And so for a long time, really nobody's been concerned with whether they're going to survive or not. That's going way back. So going back not so far, like when I got my PhD in the 70s and 80s, I became fascinated in these creatures for a number of different reasons. One of the things I studied was their circulatory system and how they get oxygen. You said we had some of these guys lying around?
In that blue cooler.
All right. I'm giving a talk here for a bunch of fifth graders or something like that a couple of years ago, and I'm like halfway through the talk and I pull out a live horseshoe crab. It was all over at that point. Just total chaos. Everybody had to hold it. Okay, which is probably the way it should be. And feel free to come up and play with these guys. Okay, so these things down here, right here, these guys, these are the gills. Okay? And they're called book gills because they're like pages of a book. Every flap has a whole bunch of these pages. And they're kind of like the plastic pages of photo books with two layers. And so blood flows through those pages and gets oxygenated as they flap their gills. Right? And so these guys breathe by flapping these gills and circulating water over them. And the oxygenated blood gets moved by the same flapping movements, pumps the blood up to their heart, which is located all along the top of their carapace here. So it's a long tubular heart. And then it enters the heart, and the heart beats and pumps it out. And those two kind of work together. Same way your atrium ventricle kind of dub, 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 dub. The gills kind of flap close, oxygenate the blood, push it up to the heart, then the heart will expand, goes in the heart, pumps it out. And they have a pretty sophisticated circulatory system. It's called an open circulatory system, but it's still pretty good. So they have this heart along here, and they have the gills, and then they have these legs. You can see the legs here moving around, and then their mouth is right there in the middle of their legs. And if you put a little bit of food in there, they start chewing like this. It's very stereotyped movements. And as we'll talk about later, we feed these guys as part of an experiment they'll talk about later. And we'll get to this, but while I'm holding him here, this male, these front two appendages, they look kind of like boxing gloves. They're like this. And that's what male holds onto the female with during the spawning season. Tail can't hurt you, despite what everybody tells you. Pick them up by the tail. It's okay to pick them up by the tail. Then there's parts of the body that we don't even know what they do. Still, like, what's that little thing, and so on and so forth. And then their eyes, right here, they have eyes all over. 2 eyes here, eyes underneath, eyes on their tail. They have eyes all over the place. But these 2 eyes, guys won a Nobel Prize back in the 60s and 70s by studying using these eyes to figure out how we detect light and so on and so forth. Right? So it's been used as a experimental animal for years because it has certain features that make it very accessible for studying basic processes. Any questions about this guy while I'm holding him?
How old is it?
This guy? I'm guessing like 10. These guys, like lobsters, they'll molt and grow every year and keep going like that. These guys will do the same thing, but after they get to a certain age and reach sexual maturity, once they become adult, they stop molting. Right? So they don't get bigger and bigger and bigger and bigger. Right? And the males stop molting. I think they'll stop molting like 1 molt short of females. Females are always bigger than males. Okay. Is there another question over here? No? Yes.
What do you mean by open circulatory system? Are you just
So you're.
So are there blood vessels?
Yes, blood vessels all over, but your arterioles connect to the vein, and yours is all connected together. Blood does exit and enter from your arterioles and stuff like that. But in these guys, when it reaches the end of the arteries, it just kind of spews out into the tissue. And then it gets, so it's open in that it's not all continuous. That makes sense?
So you don't have to see tiny little
Well, the tiny little vein vessels are not touched.
So can I give you a microphone to use? It might, given the room is so full, this actually might help a little bit.
I can talk. This is weird. This is too weird. I'll talk. Yes, should I?
If the room is full, so
It's not usually this full, so you're very popular.
Okay, I will try to use this, okay. This is what it's like to have an out-of-body experience. If anybody wants to come up and play with these afterwards, you're happy to, especially if you're under 20. So, anyway, they have this interesting circulatory system. And instead of red blood cells, they have, and hemoglobin, like you have, they have this stuff called hemocyanin. Hemoglobin, as part of the oxygen binding part of the molecule, has iron in it. Right? You've all heard of iron poor anemia, all that stuff, right? So your blood is red when it's oxygenated because of the hemoglobin. They have copper, so their blood is blue, and that's a big difference. And as we'll get to later, their blood also contains another important substance that has made it very valuable. This is you.
Great, thanks, Wayne. Chris Schwab, Plymouth State. So, I want to talk a little bit now about some of the reasons why they're so important. for the ecosystem and for we humans as well. The adults and juveniles dig to feed, and they dig especially in the muddy intertidal. Here's a female and a male that just actually came up May 9th a couple of years ago, just emerged. She and he probably spent the whole winter underneath, in probably 40 feet of water underneath the mud, and they came up to spawn. In addition, with digging to spawn at the upper intertidal, they'll also dig in the muddy intertidal. And by the end of the season, the beginning of the season, those mud flats are really very flat, but by the end of the season, they look like a moonscape. I don't know if you can see all of these pits, but that's basically every single one of those pits is feeding of a horseshoe crab, male or female. So they do something called bioturbation. They turn over the mud and bring nutrients up to the surface for a whole bunch of other organisms and allow them to survive as well. So that's a really important ecosystem service that they provide. In addition, horseshoe crabs lay lots and lots of eggs up and down the eastern seaboard, as Wynn showed you, all the way down the Yucatan Peninsula. In areas such as Cape May, New Jersey, there are millions of these animals that come in and millions of birds that come in and feed on the eggs. Now, don't feel too bad for the horseshoe crabs. The eggs, they lay about 109 tons in one area, and these birds arrive emaciated after nonstop flights of 2,000 miles or more, 40% body loss of their weight, and they fatten up over 2 weeks and then can fly on to the Arctic where they can breed. And what happens is, as these horseshoe crabs dig in the sand and the mud, they turn over more sand and bring eggs to the surface. And these animals basically eat those eggs that are on the surface, lots and lots of them. So without this food for migrating birds like the endangered red knot, it would be very problematic. And Wynn will talk a little bit more about that later on. So in the last 30 years, Wynn and I have been, 20 years, I guess, 30 years, 20 years or so, we've been studying these animals. And as most of you know, as Wynn talked a little bit about, during the spring, horseshoe crabs mate and spawn. The females come up to the beach, often with the male attached. She digs a nest, she lays her eggs, drags him over, he releases his sperm, and then they go off. And generally spawn 6 or 8 or 10 times more at other beaches for the rest of the 6 to 8 week breeding season. Males, oftentimes there's more than one male that's involved in the spawning event, and we'll show you some pictures of that a little bit later on. You can see lots of here. You can't tell who's who here, however. So more than one male can fertilize eggs, and so you'll see that when a female lays eggs, the male on the back, you might imagine, gets most of the paternity, but actually one of the what they call satellite males that are on the outside, actually have a little bit higher paternity than the male that's attached on the back. So back in the 1990s, maybe that wasn't 30 years ago, the assumptions and dogma about these animals were that they come in to breed and spawn during the spring, but then they go offshore and are tens of miles offshore during the winter. The other dogma was that they only spawn around new and full moon and high tides, only at night. And actually there's a beautifully illustrated book called Crab Moon that I remember reading to my kids, where they say that you have to get up at night to go out and see the crabs spawn. So these are kind of some of the dogma. Lastly, the dogma was, the assumptions were that these guys had circadian rhythms of behavior, which didn't really make a whole lot of sense to us because we knew they spawned according to the tides. And circadian is, circa is about a day, DN. And those are the rhythms that we all have. And horseshoe crabs have them as well, but we didn't really believe that they had circadian rhythms of behavior. So that's where we came along and started to do some work on horseshoe crab behavior.
So basically, Chris told you all those things so we could tell you that we found all those dogmas to be wrong. Okay. So we started studying the horseshoe crabs in part by putting tags on hundreds of them and then asking people to report if they'd seen animals with those tags. And we also put, let me see, where's the button up here? Yeah. You can see this is a tag we put on the side of the shell. It's a sonic tag that puts out these pings every so often. And with a hydrophone, we can hear the pings. And more recently, we've had tags like that that'll also transmit depth and activity and things like that. So we can learn a lot about their behavior while they're actually running around in the estuary. And you can see this on the slide slide over here from one of our papers. You can't read the numbers, but basically this horseshoe crab was down here in Little Bay near Newark's in the winter and moved all the way up here past Jackson Lab towards the Discovery Center and in the middle of Great Bay in the spring, stayed there for a while, and then moved back down here in the fall and winter. So this is sort of the typical movement pattern we see for these guys. They don't go all the way out to the ocean. They stay within Great Bay. They typically overwinter down in Little Bay in the deeper part of the estuary. And we also found that this movement was somewhat temperature dependent. And I think, let me see. This is a cool video. Should I jump out? Okay, so I was telling you that we could track these guys, okay? And so this is a video showing the track of one of these horseshoe crabs, a female, and showing how she moves into, oh, we'll leave you the best. Showing how she moves into shore to spawn. And this is, again, this is the middle of the night over about 7 or 8 hours. She's, oopsie, my bad. That was me.
Yep.
She's right here. Okay, that's good enough. And you can see she moved over here and then right along here. This is Jackson Lab over here, Adams Point, and she did her spawning thing right there. So with data like this, we can quantify this female spawned how many times, right, in a given spawning season, right? And data like this were also used to show that they spawn during the day high tides and the night high tides. They don't care if it's the new moon or the full moon. Females don't spawn every high tide. They'll do it like every, what, 8th or something like that. So females don't come to shore at every high tide. I think they have to recover a little bit from laying the eggs and everything else before they spawn again. Males come into shore more often.
Lynn,
Yes.
does the ping tell you how long she's there to know that she actually spawned in that spot?
This is a stupid question. We're going to ignore it. No.
Thanks.
Totally valid question. How do we know she spawned? We don't.
Okay.
So we call these apparent spawning events, right? But we're not sure that, obviously if she stays for a while. We have confirmed on a number of occasions, you guys or we will have walk on the beach and we'll make a note. Saw female number 4985 buried and spawning today. And then later on, as we're looking through the videos, we can say, oh yeah, she did spawn. So we did confirm on a number of occasions. I don't know many examples of seeing that and having a like she didn't spawn, right? But that's a very good question, thank you. Anything else?
I think it'll keep going.
All right, so as I suggested earlier, we found that many of these dogmas weren't true. They don't just spawn on the new and the full moon high tides. They don't just spawn at night, right? And so you can, these are the data here showing that they don't just spawn at night, right? So this makes you wonder, if these guys are so synced into the tides, how do they know when it's high tide? Right? So either they have a clock that's set on 12.5 hours, or they can sense the change in water depth, right? And it turns out it's both. And that and one reason that the dogma was that they spawn on new and full moons is if you've ever been down in Florida, the tides are much smaller than they are here. And the only tides that are really big enough, I think, for them to detect in Florida are the new and full moon tides. So that might be part of it. And that's sort of where that, for early studies, were done. And I think this might be this do it.
So Wynn set me up quite nicely to talk about clocks and rhythms. And basically, as we talked about earlier, they have two kinds of rhythms. One is a circadian rhythm of eye sensitivity, and Wynn and his graduate students work out with this eye cup that flash light every minute or so, and looked at the eye sensitivity. And what you see down here in this nice looking graph here, it's not dark, it's right here, is that at night their eye sensitivity is much higher than during the day. And when we put them into constant darkness, that rhythm persists, so they actually have a circadian clock, just like we all do, that helps to drive this rhythm of eye sensitivity. And what this does is it makes their eyes much more sensitive, almost a millionfold more sensitive at night. So they can see just about as well under a full moon at night as during the middle of the day. Right? They also show, and we showed this back in the first publication in 2004, that they have circadial rhythms of behavior, as we talked about a little bit. So we came up with this kind of crazy idea to put horseshoe crabs on running wheels. Put them together with 5-gallon pails. This is the bottom of one, the bottom of the other. They're facing each other, have a nice slot for the tail. And they rotate that, and every time they move around, there's a magnet that trips a switch, and so we know when they're active. And we had lots of these running wheels in buckets. And we showed that when we put these horseshoe crabs, into constant conditions, they do have those circadial rhythms. In constant water level, there's an internal clock that will drive them to become active about every 12.4 hours. And that's what these data are showing up here, these darker lines. This is activity per 5 minutes. So this is 1, 2 times per day, 3, 4, 5, 6, 7, 8 times over 4 days. That's about circadial. You guys all know, a lot of you anyway, that the tides come about twice a day. And that's what we're showing here. So as Wynn mentioned, they can also sense changes in water depth, and that helps them to synchronize with the tides out in the bay. So they have the internal clocks and the ability to sense the tide, to synchronize their activity for spawning and for feeding, by the way. So what about other populations and younger life stages? So we did a study a few years ago where some graduate student or two, this is Tyler Thomas, and I went down to Florida, and we collected animals from the east coast of Florida, Indian River Lagoon, around just south of the Cape Coast, over here St. Joe's Bay, and then of course up here in Great Bay. And the interesting thing about these 3 different populations of horseshoe crabs is that they're exposed to very different tidal frequencies. Those of us who've grown up in this area or know this area really well know that there are 2 tides per day, but other areas don't have 2 tides per day. So believe it or not, if you go into the Gulf Coast of Florida, north of Tampa, there's 1 tide per day. That's it, because of primarily the shape of the Gulf here. As well, over here on the East Coast, it's 0 tides per day. They're in the Indian River Lagoon, which has very little water flowing in and out during tidal cycles, so it's essentially a 0 tide population. So we asked the question, what what kinds of rhythms would these animals show. And we used a system to,
If you go back, if you go backwards and come to the slide again.
yeah, that's probably what I'll need to do, yeah.
Go one slide back
Yep.
and then again.
Right. So you can see these guys moving here. These are little tiny, we call them tiny juvies. They're very small. They're in very small 6-well plates. And they just hatched about 2 weeks ago, and they're moving around, and we videotape them, and then we quantify their movement. And we can record hundreds of these guys at a time, and we do, and we look at what kind of behavioral activity they have, essentially. So this program quantifies the distance moved, and they're plotted into what I'm going to show you next, which are called actograms, activity graphs, essentially, showing when they move and what their patterns are. So again, 3 different populations, 3 different tides per day. 2 tides per day, 1 tide per day, 0 tides per day. And we brought them into the lab and show you these data here. Here are Great Bay animals up here. Now, I just want to explain what this actogram shows. These are 4 days worth of data. This got a little bit screwed up, but basically this is 0 to 48, 24 to 48 hours. So on the bottom is time of day, and on the x-axis is day number. So this is day 1, 2, 3, 4, 5, 6, and so forth. It's about 20 days worth of data. And what's important here is not that you completely understand what an actogram is, but the pattern of activity that they show. And you can see here 1 bout, 2 bouts, 3 bouts, 4 bouts for 2 days. That's about 2 bouts per day of activity. That's circa tidal activity according to this northern east coast. But if you look down here in the Indian River Lagoon and in St. Joe's Bay, Florida, how many bouts do you see per day? Just 1, right, exactly. So these guys actually have a different kind of a tidal clock, which makes a lot of sense for where they grew up. And we showed very convincingly that it has mostly to do with genetics as opposed to the tides that they're exposed to. Because what we did is we collected eggs from all 3 of these sites, freshly laid eggs that within 20 minutes of them being hatched, put them in buckets, flew them home to Plymouth, New Hampshire, and put them into 3 different conditions, either the 0 tide, 1 tide per day, or 2 tides per day. And the conditions that we gave them didn't matter much. It was their genetics that seemed to matter a great deal.
Does the slope of the graph signify how the tides are changing daily?
It really does, yeah. So this is about a 12.4 hour period. Absolutely, good eye, yeah.
Just a curiosity question. So if you move back with a 0 tide to a 2 tide 0, he will not survive there?
Yeah, we wouldn't do that in the field, but we did that in the lab. They did perfectly fine, but they only showed when you take a 1 tide per day animal and you put it into a 2 tide, they're just going to exhibit 1 bout of activity per day. It's an interesting question. Yeah. So point here is that the different populations up and down the seaboard here have different genetics. And as well, we can use these tiny little guys that we hatch from eggs to really help us answer a lot of important basic biology questions. In fact, we released about 200 today from the winter that hatched last year out in Great Bay. Another question?
So does it make them different species? Or they do the
Yeah.
different angle at all? Or?
So different populations. They certainly, at this point, we would call them the same species, but give them 1 million years, and we'll see. That is an experiment we're running right now. Okay, that's a good question. Yeah. So they seem to be the same species, but definitely different populations. So it wouldn't be ethical for us to take animals and transplant them down. They do fine, likely, but it's not a good idea to do that. That brings us to the next segment, whether they are on
Whatever
decline.
that is. No, it's an interesting question. How many subpopulations are there? Right? Because with the genetic tools we have these days, it's very easy to do the genetics and try to figure that out, how much mixing is occurring. So let's rewind about 30 to 40 years. And people down in Chesapeake Bay area, the Audubon bird people, are noticing there's some issues. The bird populations are declining a little bit. The birds aren't looking good. And so they started raising some warning flags, and the people down there in charge of keeping track of the horseshoe crabs and whatnot for the state started looking into that, into the situation. And basically, the bottom line is that the horseshoe crab populations were declining significantly, and oddly, the first people to notice it were the Audubon people, because nobody was doing horseshoe crab surveys back in those days because there were so many of them, right? And one book that was written on this whole story, The Narrow Edge, by Deborah Kramer, I highly recommend it. She came to visit us, talked to us, and she really is a smart woman and has her act together. And she did a very good job of describing this whole situation. And as you can see here, this shows catch per unit effort, right? In these surveys they do from 1990 till 2005. And here's that decline I was telling you about, right? So the numbers were plummeting, and people were beginning to get very worried about this, right? Why were they declining? Well, there's two major reasons. One was that some fellows at Woods Hole, Fred Bang amongst them, discovered that there was a substance in their blood that was very sensitive to gram-negative bacteria. So it was kind of like their immune system, or your white blood cells, right? And you could extract this substance that became called limulus amebocyte lysate, and you could mix it with, before you got a blood transfusion, mix it with a vaccine, whatever. You could test substances that were about to be put into your body at the hospital to see if they were contaminated before you did so. They used to do this by injecting rabbits with these and seeing if the rabbits would respond. So you can imagine how expensive and time-consuming and stuff that was. So this was quite a discovery. Plus this LAL stuff was very, very sensitive, right? And so literally now, everything that goes into your body gets tested with this. And you can imagine during COVID, with all the vaccines that are being produced, really a huge need for this material, right? They get the material by bleeding these horseshoe crabs, shown here, and they remove about 30% of their blood volume. Now, all that blood you see there is not made of LAL. Mostly that's regular blood full of that blue hemocyanin stuff I told you about, right? And just like when you give blood, if you've ever seen the tube, right, an hour later, there's a little layer of white stuff on the top, that's your white blood cells. That would be like this. So you need a lot of blood to make a little bit of LAL. Right? So every year they bleed, 600,000, 500,000 horseshoe crabs to get this stuff. And a certain percentage of them die, 20, 30% of them die. They like to bleed the females more because females are bigger and have more blood. And then they release them back into the ocean. And maybe more die, we're not sure. Right? And so Chris and I started looking into this. And we would subject horseshoe crabs to the same bleeding procedure at the Jackson lab. We put these little accelerometers on that are kind of like, same accelerometers you have in your phones that tell you how many steps you took in a day. And then we would let them run around, and we bleed some, and we wouldn't bleed some, and so on and so forth. And we found out that after you bled them, it took a week or two for them to recover their same level of activity. They were lethargic for a while afterwards. Right? Okay. And a certain percentage of them died, as you might expect. Right? Fewer of them died in our hands than at the bleeding companies, because when you bleed them, they have to pick them up on the boat, put them on the deck of the boat, to drive them to the factory, let them sit overnight, bleed them. It's a rough process, in addition to losing that blood, okay? But we also noticed, we also thought when we did this, we wondered if we released them back into their natural habitat, would they also behave differently, right? Would they recover? And would this affect them during the spawning season so they would spawn less? Right? Because if they're spawning less, this could have a big impact on the population as a whole. So we bled females. We collected them early in the spawning season. We bled some. We didn't bleed others. We put those little tracking devices on them, like I showed you earlier. This shows you an example of a track from one of these females. And we found that the bled females subsequently mated about half as much as the control females, or expressed apparent mating events half as much as the controls. We did, and Megan Owings, one of my graduate students, did this in 2020, and then we repeated it again and got the same data. So this is something to be concerned about. Okay. The other way that we lose horseshoe crabs is as bait for whelks. So they capture horseshoe crabs, they cut them in half, they put pieces of horseshoe crab in the bait bag, and they fish for whelks. And we lose, on the right is a graph of, these are horseshoe crabs harvested and used. So the green is commercial, is bait landings, and you can see right around now, it's averaging about 1 million horseshoe crabs per year for the bait people. Now, all of those guys die, right? Okay? So you have 1 million that are dying from that, and then this black one shows, the dark green shows harvested for biomedical, and then the black bars are estimated mortalities from that, right? So the combination of these two phenomena, starting 40 years ago, has led to the demise of a number of horseshoe crabs, right? And this, as I showed you the data earlier, this is what led to that decline that people started documenting down in the middle of the country. So one of the things we're doing now to try to help is to develop an alternative bait for whelks. I'm doing this in collaboration with a number of different people, including Liz Fairchild, you might know her, she's a professor at UNH. One of the baits we're using for, there's a couple different places we get alternatives. One is oyster mussel factories, they collect them all, they bring them in, and they drop a bunch on the floor. We get those. Right? Kind of like the way you used to get pig food at the through a marketplace, because everything they dropped on the floor they had to get rid of. Right? So these sort of waste bivalves, but we also take green crabs that are sort of an invasive species now. They're everywhere. We take those and make them into bait. We also take the excess horseshoe crab blood from factories, the stuff they don't use, the blue stuff, and we mix that in with it, and so on and so forth. We make these little bricks of fake bait, and we use that, and we've had some good success with that. So we're very optimistic that that might eventually some commercial entity will start making that on a big scale, and it'll be cost-effective alternative, so they don't have to kill the horseshoe crabs.
Where is this whelk fishery?
A lot of it's like we collaborate with people in Massachusetts. Like there's a big fishery, Martha's Vineyard area, right, and then down just all the way down to South Carolina, kind of thing.
And the whelks are used for, oh boy.
Yeah. I've never.
Follow the sauce.
I keep meaning to try some, right? And I had some whelks, some conch soup the other day just to try it, but of course it wasn't. It wasn't the real thing. I need to try the real thing, but most of it is probably going to Asia, where it's a sort of a delicacy.
Well, is anybody working on trying to get the labs to move the collection process closer to where the horseshoe crabs are? I know, I ask all the important.
No, it's, can someone escort this woman out of the room?